Today we are celebrating the 100th, well, not today exactly, but we're here today to celebrate the 100th anniversary of the birth of Wallace Coulter. Wallace was perhaps one of the original biomedical engineers. He was an engineer, but he was also very translationally focused. He always wanted to do things to better mankind. I believe the motto of the Coulter Corporation was "Science Serving Humanity." After Wallace passed away, many of the proceeds of the sale to Beckman Corporation went on to form the Wallace H. Coulter Foundation, who we've been interacting with now for about 8 years, and it's been quite a journey for us. The foundation has been extraordinarily generous to us, both in terms of their time and their finances. They've been demanding at times, but always generous, and never failingly trying to make us better at what we're doing, never failingly trying to keep us focused on the task of moving technologies to the patient. There are lots of pilot programs for technologies and medical science around campus, but none quite like the one that was done here. There's a number of unique aspects to that. One of those was the focus on bringing technologies to the patient via a commercialization pathway. Another was the instituting the oversight committee that involved a diversity of people, including clinicians, venture capitalists, people with industry experience. More than half the people in our oversight committee are outside the university, and I think ultimately that proved to be a very wise decision, because having served on internal and external pilot selection committees, I really appreciate the kind of the outside view, the not swayed by institutional politics. The stature of someone within the institution doesn't matter. What matters is the technology and whether or not there's a pathway to bring it out. So that's a very unique aspect of the program that I think has been very successful for us. Another unique aspect is the appointment of the Coulter program director. That's a person whose job it is essentially to stay focused on our projects, to help each of our projects, to find projects that are suitable for the program, but also to focus the projects, help guide them through the various stages, to try to keep them on their milestones, kind of once they graduate from funding, to help them find their way out to the next step, whether it's venture funding or a license, and trying to keep the technology moving forward at all times. We often say once a Coulter project, always a Coulter project, and so even those projects that have ended a couple years ago, we periodically come back to with our Coulter program director and take a look at those and say, is this one still have life? Is there some way that we can move this project forward? And so we keep an eye out for those. And those are a number of the unique aspects of the program. It's been a team effort from the beginning. First of all, the faculty, the clinical collaborators, of course, are a key part. There's no success unless there's great ideas coming from our faculty. The oversight committee is past members and current members. A lot of people have put sweat into this, have diligently come to our oversight committee meetings well read and well prepared and ready to discuss the projects. That's always been great to see people give up their time in that way. Our Coulter program directors, we've had a number of good ones over the years. It started with Chris Alto a number of years ago. Then Jim O'Connell served in this role for a while. We have Matt Bell sitting in the back of the room there, who was one of our most recent ones. And then our newly hired Coulter program director is Tom Martin. Tom, if you can just give a little wave there or say hello. So Tom will be starting officially in this job on November 1, and I'm very anxious to see great things follow after this as well. And so our Coulter program directors have been a key cog along the way, and I'm really happy we've had such a talented group. The team involved the Office of Technology Transfer, essentially at all levels. Oversight committee meetings, our review selection meetings, there's been at times maybe as many as Well, certainly over the course of the meeting, there's been as many as 10 people in and out of the room from the tech transfer office who've offered things there. And then certainly after the projects kind of are graduating to the next stage via licensing or the business development office, Technology Transfer has been a great partner in this endeavor, and I certainly appreciate everything that they've done. I can't forget the foundation themselves. They've been very active at every step along the way and have given us advice on how to, both in terms of participating on a very regular basis, but also, for many years, and is continuing. There's a sort of at times biannual, at times annual pilgrimage to Florida to meet other schools that have these programs and learn best practices and turn the microscopes on ourselves in our programs, and that's really been an extraordinarily valuable experience, and as well as been a great networking experience for me as well. And finally, I want to acknowledge the local biotechnology community who've been very active, members of our oversight committee, as consultants, as investors into our technologies. They've, essentially it's the whole ecosystem that have made this work. And so this was really the brainchild of the Coulter Foundation. They've learned as we've moved along and we've been learning with them, and hopefully we keep getting better and better. So next I would actually like to introduce Dean Jim Woolscroft from the medical school, who's going to make a few opening comments.
It's really a privilege to be here. First of all, I too want to thank the Coulter Foundation for the tremendous impact they've had. It's interesting, some of you, like me, are clinicians, and so understand what a revolution this gentleman created with his contributions to clinical care in cell counting, flow cytometry, really changed the way medicine is practiced and impacted hundreds and hundreds of thousands of people every year. And when you count up the number of years, it's probably hundreds of millions. And so in a way, that's the goal going forward. How can that legacy be promulgated? As Doug outlined, the Coulter Foundation and our interaction here at the University of Michigan with them has already had a very significant impact on the university. I'm sure many of you are aware the many programs that were funded that have already led to startups and major programs going forward. Histosonics, a really innovative use of ultrasound technology, is in clinical trials, tissue regenerative systems, INSEP, 3D biometrics, and others. But very importantly is the impact it's having going forward. We learned a great deal from the process, as Doug has already outlined. Many of you know that we were successful in competing for a $2.4 million grant from the Michigan Economic Development Corporation earlier this year. The components of that grant were built upon the lessons we learned from our interactions with the Coulter Foundation, with advisory committees, working together across the university. This grant has been augmented with an additional $5.1 million from the Dean's Office in the medical school, from the departments in the medical school, from the Office of the Vice President for Research, for a total of $7.5 million over the next 3 years. We call it MTRAC. It's really part of the strategic plan to move forward innovations in the life sciences. Very interestingly, we had an initial call for proposal, and there were 39 proposals received. As has been the experience with the Coulter, we've put together an advisory group to review them, that includes many outside members, serial entrepreneurs, people that have developed and taken to market innovations, in addition to internal people to review these proposals. And we'll be announcing the winners in January. We've also really formalized in the medical school our commitment to innovation. Just a couple of days ago, we announced Kevin Ward has agreed to be our Director of Innovation. We spent a great deal of time and effort over the last 2 to 3 years facilitating the work of our basic scientists through cores, through seminars, just a wide range of things. And now going forward, we have a similar commitment to translational and clinical research. And so we're looking to Kevin to help lead in a four-pronged approach, looking at therapeutics, looking at diagnostics, looking at devices, looking at health IT. How do we ramp up, take the great ideas that our faculty have, move them forward? And much of what we're doing is predicated upon the lessons learned from the Coulter Foundation collaboration. In addition, something that I am incredibly pleased that we've moved forward is the joint biomedical engineering department between the medical school and the College of Engineering. And Doug, before he went off on this vacation, which he calls a sabbatical, was the inaugural chair of that. And I have to give him and his faculty tremendous credit for being willing to risk, visionary enough to come together in a very different way. There's tremendous excitement on the medical school side. Already there are 2 faculty that have been hired with their roots in the medical school. And we look to this collaboration as another outgrowth of the collaboration that started in large part and was catalyzed around the Coulter Foundation. Can only imagine what will be forthcoming from all these efforts. But we have great expectations for the future based upon the quality of our faculty, based upon the collaborations that have been initiated and what will be forthcoming. At this point, it gives me great pleasure to introduce Mr. Wayne Barlin, the Vice President and General Counsel of the Coulter Foundation. Mr. Barlin.
Wallace Henry Coulter was born on February 17, 1913, and he died on August 7, 1998. His headstone simply reads, Dreamer, Innovator, Entrepreneur. But there is so much more to the man than just what he did, and I'm not just talking about Coulter Corporation. I'm talking about Wallace. He was part of the greatest generation, and they were notorious for not bringing attention to themselves. That is why I am here today to tell you more about Wallace. I had the pleasure of working with Wallace for 20 years as General Counsel of Coulter Corporation. And before I go on, since a picture is worth 1,000 words, I'm going to show an 11-minute video about Wallace.
Wallace Coulter grew up during the era of invention. He was a thinker, a tinkerer, a dreamer, and a born engineer. He will always be one of the great inventors, innovators, and entrepreneurs the world will never fully know. Now, 100 years after his birth and more than a decade after his death, this American original can teach us all lessons of loyalty, inspiration, tenacity, and spirit. As CFO of Coulter Corporation and later trustee of his foundation, I pledge that Wallace Coulter's legacy will endure for future generations.
If in today's world, with all these smartphones and the iPads and everything else, if somebody said to you, a guy in Chicago has developed something that you pass a particle through a hole in a jewel in an electric field and it generates a pulse and we can tell how big it is, they'd probably say, what's the ball game score? It would probably just be insignificant. But you have to go back to those days to realize what we had, and we had nothing in comparison to it. And it was monumental in its time.
Wallace's inventions had a tremendous impact on healthcare and on the quality of life, really, for the world. And it was really that automated cell analysis, those first machines, that launched the idea of having this type of routine diagnostic information available.
Many patients are alive today and cured with their cancers and with their autoimmune and other diseases because of the foresight and the vision that Wallace Coulter had, not only for science, but to make the science count for patients as well.
That the world is better off because he was in it. And I'm not simply talking about the thousands of employees that had their livelihood from the Coulter Corporation. I'm talking about the literally billions of people that have better health because of the things that came out of his head.
You had to have grown up in the period where everything we did was manual to have a real appreciation for the impact that those instruments had on laboratories.
Wallace's invention came in his basement when he was actually working on something else, but nevertheless, this was the invention which changed hematology analysis completely.
The Coulter principle, it's a straight electronic principle. We can count and we can size any particle that you can get suspended into electrolyte, and simultaneously we pass a current from one electrode to another through an aperture. It's like a little cylinder. Now when this particle enters that sensing zone, there's an impedance, and a pulse comes on an oscilloscope. The height of the pulse is proportional to the volume of that particle. That's what opened up research around the world. You can only get 2 dimensions on a microscope. Coulter's electronic principle is cubic volume. We can count and we can size, thanks to Wallace Coulter.
This is one of the very simple inventions that was really a great breakthrough, and the impact just cannot be measured. It's almost like inventing the wheel, something that you couldn't dream doing without.
It's an invention that keeps on giving, and Wallace should be very pleased to watch the fruits of his invention blossom throughout the world.
He liked to make things simple. He liked to get to the essence of things. And in a sense, Wallace was like a beachcomber. He had a knack for spotting ideas, even if they were in some obscure form.
He was inventive, but I think his greatest gifts were seeing how to make an invention practically useful.
Nowadays, we talk about personalized medicine. It's really the buzzword. It's the right medicine for the right patient at the right time. Because of Wallace Coulter's vision, personalized medicine really started several decades ago. Because personalized medicine predicated upon the fingerprint of an individual patient's cancer was started because of his vision. I think the best way to think about it is it's a before and after Wallace Coulter, and the paradigm of treating and understanding disease was completely revolutionized as a result of his brilliance and his prescient forethought.
You know, and you imagine the kids that have been diagnosed with leukemia, lymphoma, all right, the anemias, yeah, megaloblastic or iron deficiency, thalassemias, aplastic anemias, all of these things, we're all going through a Coulter Counter.
Not only in the biological end of the world, but what we call defined particle or industrial particles like pigment in paint for quality control, chocolate, moon dust that was counted on the Coulter Counter, for instance.
So the Coulter Counter was just the beginning. It established the principles that a lot of other technology grew from, and that includes the flow cytometry.
And the impact that that had is unbelievable, because I think that it was the beginning of the revolution in medical treatment.
Wallace had a vision for what he wanted to accomplish, what he wanted to impact, and his vision was big. It was really big, and for as much as he accomplished, I doubt that he feels like he maybe got 10% of his vision in his lifetime, really.
He wanted to bring something new continually, but even when he achieved that success, he didn't stop. He carried on. It was the next challenge, the next thing. And some challenges fail, some challenges succeed, but he would never stop.
I think he was innately enterprising. He had that entrepreneurial spirit.
For me, an entrepreneur is somebody like Wallace. He had a dream, he had an idea. He was willing to risk the company to do his idea.
Yes, he took risks, but he had such a great analytical mind that he took calculated risks and won most of the time.
Wallace Coulter is probably a unique entrepreneur. I don't know too many who combine this excellent access to scientific knowledge combined with the ability to translate it into action.
He was a prime example of someone who, in fact, made something from nothing. He didn't take technology that was already existing and tweak it and make it a little bit better and sell that. No, in fact, he invented product.
All of those companies, which today has an important position in this field, they are all seeded by Wallace's innovation. And not only the technology, but the people. And they all go to all different companies. They carrying forward with Wallace's mission to help people's daily life.
ウォルス・ヘンリー・コルタさんはいずれにしまして我々の業界においてもまさに先駆者でありますし。
Wallace H. Coulter was a pioneer in the industry. He had tremendous insight and passion for technological advances, as well as enthusiasm for business. It is something we admire regardless of our business relationship, whether we are competitors or not.
So for us at Coulter, it was always about science serving humanity. And what does that mean? What that means is that at the end of the line, there is a patient. And that's what it's all about.
I don't believe they were ever really interested in the money. I think they were interested in creating value.
I guess we had to make money, but I do remember one conversation with Wallace one day about profit. And Wallace explained to me what profit was. He said profit is the very small difference between two very big numbers. You got the sense that the reason we were bringing dollars in was to invest those dollars back to carry things on, to go the next step, to get the next thing done.
I tend to think of Wally in a way as the Christopher Columbus of size analysis. He took us into uncharted waters and gave us a view of the new world.
His legacy is that small beginnings can grow into great things. The invention of the Coulter Counter, the Coulter principle, is so elegant. And so what appears, once you've explained it to somebody, they go, ah, well, that's obvious. But it wasn't obvious. It's a very elegant solution to a very important problem.
He clearly gave patients and caregivers alike the gift of hope and ultimately the gift of life. And there is no greater gift than that.
Wallace's enduring legacy lives on in the people that he touched, taught, and inspired. All he ever wanted to do was to serve humanity, and he did.
Wallace was slow of speech and lightning fast of mind. He was mischievous and inquisitive. He was kind and often had a twinkle in his eye. He came from humble beginnings. He was born in Little Rock, Arkansas, and grew up in McGehee, Arkansas, 8 miles from the river. His father was a railroad dispatcher, and his mother was a kindergarten teacher and a member of the Daughters of the American Revolution. That instilled in Wallace a very patriotic spirit. He was a child of the Depression. His humble surroundings became the foundation of how Wallace lived and how he lived his life. Wallace was a voracious reader. Reading was the way he fueled his curiosity. His world became infinite through the books that he read. They were influential to his life and his career. He often gave books to people as presents. He often gave books as presents to people who were close to him. He played both basketball and football, but he was smaller than his classmates, having graduated high school early at 16. He went to college at Westminster and then transferred to Georgia Tech to study electrical engineering, but he had to leave after his third year. It was the Great Depression, and he didn't have the funds to continue. Early on, he worked in Memphis, Tennessee, as both a radio engineer and announcer. He went to work for General Electric in 1937 and jumped at the chance when the opportunity came to work in Asia as a GE sales and service engineer. In February 1942, when World War II came to where Wallace was in Singapore, he escaped by boat and took the circuitous route back through India, around the African continent, through South America, and home in time for Christmas. He could not go home through Europe because the war was raging. At each port of call, he would stop into the General Electric office and have his expenses paid. What a way to see the world. Finally, he arrived home in time for Christmas. He moved back to Chicago after the war and worked for several companies, including Raytheon and Peerless Wireless, all the while maintaining a research lab in Chicago. And that is where the Coulter principle was invented, in a basement research lab. He applied for a patent in 1949, and it took 5 years for the US Patent Office to grant it because they kept on saying, you can't patent a hole. Soon after, he founded Coulter Electronics, which is the precursor of Coulter Corporation. Later, Wallace was a founding member of the International Committee for the Standardization of Hematology. Once a meeting was held in England at a castle. Upon check-in, Wallace was given a room without a bathroom. The bathroom was down the hall. While everyone else attending the meeting had a room with a private bath, Wallace never complained, though in fact he was paying for the meeting. And it was not until one of the attendees' wives found out and told hotel management about this did anyone know. Always humble, his middle managers lived in bigger houses and drove newer cars than he did. His castle was his R&D laboratory, where no expense was spared. In fact, I'm going to digress here for a minute. In December of 1998, I had to get his signature on a tax return, and there was one section where he had to sign the tax return. But he wasn't in the office that day, so I had to go to his house. So I went to his house, and they called ahead and said I was coming over. He didn't know me at the time. And I got there, and he was very nice. He signed every place I told him, and I went in to the post office and had the return timely filed. And then when I went home, I mentioned to my wife that I just went to Wallace Coulter's house. And the first thing she said to me was, well, what was his house like? And I looked around and I said, not as nice as this house. Wallace never married or had children. His employees were his family. He encouraged his family members and his employees' family members to work at the company. Well, for 20 years, everything ran very smoothly. The company never needed a layoff. But coming into a financial bind, Sue Van proposed a layoff of 400 people. Wallace sent her away to do her due diligence. Was the employee married? Did they have children? After 2 weeks, Sue returned with the information. The list was whittled down, but she was sent away to find out more. Did they have a mortgage? Were any of their children in college? Were they supporting people back home? You see, we had many Cuban immigrants who worked at the company. Sue returned again. After several meetings, Sue was able to convince Wallace to lay off 50 people. Well, a couple of weeks later, Sue was walking down the street. You had to walk, we were in a series of warehouses, and you had to walk from one building to another to get to the different departments. And she saw someone who she knew was laid off. The following week, she saw someone else who she knew was laid off. So she went to HR, and she went in and asked about it, and everyone began to laugh. It seems that Wallace hired all of those people back, and he gave them a raise. And furthermore, he let them keep their severance package. Science serving humanity was the philosophy he lived by and how he ran the company and how he lived his life. Wallace invested heavily in many skunk works. After the discovery of monoclonal antibodies in the late 1970s, Wallace believed that they were the magic bullet and as such could cure cancer. He invested heavily in one project using an anti-CD20 monoclonal antibody as a therapeutic agent for non-Hodgkin's lymphoma. Sue tried to kill this project many times. We were a diagnostic company, not a pharmaceutical company. We did not have the expertise or the deep pockets to continue with such a project. One day, Sue was called to a conference room with several doctors present, and I think, Dr. Kaminsky, you were there. Well, Sue knew she was being set up, but she just didn't know how she was being set up. After making this presentation about the wonderful results from this anti-CD20 product, Wallace turned to her and said, "People are dying every day. So what do you think Sue said? How much money do you need, Wallace?" After that, Sue never cut Wallace's R&D budget again, and she found a way to get the drug out to market by spinning a company out a company from the Coulter Electronics. This therapy is now used as a standard of care for non-Hodgkin's lymphoma. He spent money flaming the fruits of his passion, pushing technology, getting better answers, faster for patients and practitioners. Wallace's philanthropy started with his family, that is, his employees, giving them personal loans, helping with mortgages, paying for education, and experimental medical care. During the sale of the company to Beckman, Wallace was in the hospital. Sue would update Wallace daily on the progress of the negotiations and would ask if there was anything special that he wanted. His only request was to take care of my people. On the sale of the company, $100 million was left on the table. He made sure that every employee received $1,000 for every year of service, no matter what company, what country, or what position they held. Some received more than they made in a year. The foundation then became Wallace's family. Upon his death, the foundation inherited three things: his tropical fruit farm, his jade collection, and we also got his wealth, which enables us to carry on the mission of science serving humanity. The foundation partnered with the University of Michigan in 2005 as we began our translational research partnership. The goal of this program was and continues to be bringing industry practices into the university to accelerate the translation of promising technology to improve patient care. We have watched with pride as these programs have attracted follow-on funding from venture capital, industry, and additional government grants. We invested $55 million in the program to date, and the return at this point is nearly 10 to 1. The University of Michigan received in excess of $4.5 million in grant funding during the course of the program. As a result of the university's success, the foundation provided the university with a $10 million endowment in 2011. The university has matched that endowment to create a $20 million fund for the continuation of the program. 9 of 10 schools in the first phase of the program are continuing, and 6 new schools are participating in translational partnership 2. Also, somewhat unique to the University of Michigan, the foundation has funded the Lupus Foundation of America to provide research funding for this very complicated disease. Selection for this program was done in a similar manner to the Coulter Translational Research Award. 4 research proposals were selected for funding. One to Dr. Bruce Richardson from the Department of Internal Medicine at the University of Michigan, who I believe is here today. So the foundation has a multifaceted connection with the University of Michigan. In 1988, Wallace and Sue went to look for a site to begin manufacturing reagents for Coulter instruments. During that trip, he was asked to speak to a few students who were studying English. Surprisingly, he said yes. Sue looked at him and said, "Wallace, you hate to speak." "No problem," was his response. They were picked up at a hotel and driven to what looked like a warehouse. I'm having page problems here. They walked down a hall, and with light coming through the doorway, Sue could see that there were hundreds of people inside what was, again, a warehouse filled with bleachers. "Ha," Sue said, "serves you right." All he said was no problem. They sat in the front row, and when Wallace was introduced, he slowly walked up to the podium and simply said, "Thank you for asking me here, ladies and gentlemen. I would like to introduce you to one of your own, my CFO, Sue Van." So he's motioning her to come up to the stage, and she's going, "No, no, no." And he finally comes and he sits down next to her and he says, "I finally got even with you for cutting my R&D budgets." Sue said to him, "Wallace, you're embarrassing us. Go up and talk." And with all seriousness, he looked at her and said, "Then you go speak for me." To this day, that is what Sue is doing through the Foundation's programs and grants. So I am fortunate to be here today to be speaking for Sue. And at this point, we have one more presentation, which I'd like to ask Dean Williscroft and Doug to help with. This is a medallion of the Coulter principle. It's actually called the Galvano, and of course this is the microscopic.
Thank you so much, Wayne. It's always great to hear the stories of Wallace the person, and they are great stories. Okay, so moving on with the program, I'd like to introduce Chris Gibbons, who is the president of Histosonics Corporation, and she's here to tell you a little bit about the part of the impact of our program here, and they are Histosonics is one of our great success stories, and I think represents what we would like to do with this program. So.
Well, thank you, Doug. I really appreciate the invitation to talk today. It's a great honor to be here, and of course, it's a real tough act to follow, Wayne. That was a beautiful celebration to Wallace's life, and we hope that we can do him honor in terms of the work that we're doing at Histosonics. So I'd like to be here today to thank the foundation as well for their financial support and guidance. I think the Coulter program has given us a lot of great guidance in terms of how the technology came to the point of actually being able to be launched. I mean, it was about 5 years ago, actually, today, that I had the great honor to start an executive in residence program here in town, working with Jim Addix at Venture Investors, looking for promising technologies that were potentially venture fundable for the fund, and that may, ultimately allow me to join as part of the management team. And my very first meeting was with Jim O'Connell, and he told me about some great work that was going on at the university with the Coulter program, and told me about this technology that used therapeutic ultrasound and non-invasively treating tissue without heat, and I thought, pretty cool. Then he introduced me to Tom Davison, who was looking and had been working with the University Tech Transfer Office, was brought in also with the support of Coulter to look at portfolio companies there and to specifically help the Histosonics team, or the histotrypsy team at the time, really decide what was the best first application and how to actually get this company launched. And he was very excited, just as Jim O'Connell was. And then he took me to a dog lab. And there they were doing a procedure where we were able to see the bubble cloud. The bubble cloud here that you could see is basically the ability for all of the pressure waves targeted at that tissue non-invasively in a canine prostate, breaking up that tissue in real time. And you could see the ability to do this non-invasively with image guidance, such that you're able to treat, in this case, this canine prostate, and be able to get the tissue effect that you're looking for non-thermally. And got very excited about the opportunity, and continued on with the project with Jim O'Connell. The team came together at the university. There was Charles Kane was really the godfather of histotrypsy work. He was the one that said, if we can control cavitation rather than trying to avoid it in the application of therapeutic ultrasound, maybe that would be this great new application of this therapeutic ultrasound modality. So he, with the other members of the biomedical engineering group, really worked on this technology, and then teamed with Dr. Will Roberts from the Department of Urology, and really applied this application in the first urologic applications. This group really saw the promise and came together to bring this funding potential together with Tom Davison being added to the team. And we saw then the need to really bring, obviously, financial investors in. The team that you see on the left is the group that I just mentioned. Charles Kane with the group from Biomedical Engineering, including Jen Zhu, Brian Folks, and Tim Hall, with Dr. Roberts serving then as the clinician that they partnered with. Lots of folks, though, also came together for the histotrypsy group. And so here is the group then, as we continued to progress in terms of formation of the company. Tom Davison was working with Jim Berlina as a mentor in residence. I was working with Jim Addix as an executive in residence. And we really needed then to continue to finalize the business plan to make this venture-worthy deal. So what became very important to us, to the team, was really what was it that was going to get this over the hump to make it venture fundable. And that's, again, where Coulter came into play. This was in 2008, and for those of you that were certainly following the financial markets at the time, this was a tough time to get a deal funded. And so there's a lot of interaction with various venture funds in town that said, what is it that are the key areas that need to be funded, or that need to advance to be able to get this deal funded. So the Coulter team actually advised the group and said, with the input from venture capitalists, was key work needing to be done in the area of the IP portfolio, freedom to operate evaluation, IP analysis, and a regulatory affairs assessment. We also had done there was also additional preclinical work that was funded by the Coulter project. And you can see by this was the last quarterly report that the histotrypsy team had provided to Coulter, where all of these activities got done. And then the key milestone here being that $11 million Series A financing was put together to get the company launched and the deal done. Again, one of the key things, I'd actually asked Charles Kane, I said that I'd be speaking today, and I said, what are one of the key points that you think is important to make about Coulter's impact on Histosonics? And he said, the big team, which is the term that we use for the scientific founders together with the initial founding team from the company. And this team actually continues to meet. We've been meeting pretty much every other week for the last 4 years on working with issues that have been facing the company, technological, clinical, et cetera. And they continue to provide great advice and guidance. But it was really the Coulter funding, the Coulter advice and counsel that brought this team together. And so I think Charles's words were quite profound in terms of Coulter's impact in terms of bringing this team together. The team continued to grow after we got it launched, where we added certainly the board of directors. Tom Davison joined the company as chairman and CEO. I ended up joining the company as president and chief operating officer. And Jim Berlina, who had served as a mentor in residence at the university, joined as our CTO. The venture funds have different positions on the board, and we were very fortunate that Andy von Eschenbach, the former commissioner of the FDA, joined the company early on as an outside board member. So Histosonics, just kind of for those of you who may not know us, we have a non-invasive image-guided therapeutic ultrasound. Our first application is the treatment of benign prostatic hyperplasia, although it is a platform technology, so there's numerous clinical applications. BPH alone represents a $500 million market potential, with about $2 billion market potential overall when you look at the advanced energy space in general. We've got a strong intellectual property portfolio and world-class management board and advisors. And just a moment here to tell you a little bit about the condition that we're addressing, because again, Wallace Coulter's vision is to talk about patient care and what is it that we're doing in terms of addressing an unmet clinical need. BPH is a very prevalent condition. It's the naturally occurring enlargement of the prostate, making it difficult to urinate or giving you the sense of frequency or urgency in terms of urination, and it severely impacts day-to-day life. It's quite prevalent in that it affects half of the men by the time they reach age 50 and 80% by the time they're 80. So much so that 2 million men in the US and 4 million men worldwide seek treatment each year. And it really does represent pretty broad market potential, $5 billion in total, but the bulk of that is really in the drug therapy space. There's a lot of technologies and therapies that are coming to address this need. Drug therapies certainly work for a period of time often, but oftentimes it also postpones a definitive treatment. And there's also, it's expensive with adverse side effects. Oftentimes patients then need to seek surgical intervention, and there's minimally invasive therapies that oftentimes have delayed relief, poor durability. The gold standard, which is a transurethral resection of the prostate, is the gold standard because it actually debulks tissue and provides long-term relief, but it does come with it an 18% incidences of side effects. So we believe that the Vortex RX and Histotrypsy provides the solution to that unmet clinical need because we can offer a definitive therapy. We debulk tissue with Histotrypsy like a TERP, providing immediate and long-term relief. It's non-invasive, so it's attractive to patients, and it's non-thermal. Most all of the minimally invasive therapies are thermal therapies. So we believe the non-thermal approach will provide significantly less trauma and inflammation and faster healing, less scar tissue, et cetera. We've also found that there's much less bleeding with Histotrypsy, and we believe because of the non-invasive nature, this can be done in a urology office setting, which is certainly a beneficial approach from a reimbursement perspective. We've finished our development. This is the device. We have 6 systems built and tested. 3 have been deployed to sites. We have both an IDE approval for investigational device exemption from the FDA to do clinical trials in the US, and the equivalent in Canada, an ITA, to do a study there as well. We've got 3 sites approved on their ethics and IRB boards, and our pilot studies, clinically, our pilot study is underway. So those of you in the audience who may know somebody that's suffering from BPH symptoms, this is our way to try to attract patients. We're in the midst of recruiting right now. We have treated our first patients, and we're looking for more. So if you're suffering from BPH symptoms or know somebody that is, that's where to call. The University of Michigan is one of our sites, and we're looking to recruit today. So again, I want to thank Wallace Coulter for his vision. The translation to patient care is something that we hold very dear. We believe that Histotrypsy can really help improve patient outcomes, and we hope we do the Coulter Foundation proud in terms of the funding and the direction that we've taken Histosonics. So thank you.
Now I'd like to introduce our keynote speaker for this celebration. This is Professor Paul Robinson from Purdue University. Professor Robinson is the SVM Professor of Cytomics in the School of Veterinary Medicine and also Professor of Biomedical Engineering at Purdue. He got his PhD in immunopathology from University of New South Wales, and later he came to the US and actually did his postdoc in Peter Ward's lab here at the University of Michigan. After that time, he got recruited to Purdue, much to our loss, and there's an interesting story that goes along with that, and you can perhaps ask him about that later. And he's been at Purdue ever since. He's won numerous awards. He's a fellow of the American Institute of Medicine and Biological Engineering. He received the Pfizer Award for Innovative Research, the Gamma Delta Sigma Award for Merit Research. He's worked closely with the Coulter Corporation for many years and got an actually very early start to his research program through the generosity of Wallace Coulter. I'll probably hear a little bit about that in a second. One thing that I don't get to say about most of the speakers I introduced is actually summited Mount Everest, and that was part of an effort to publicize the need for early detection and for methods for low-cost methods of detection of HIV in the developing world, and he's founded a charity called Cytometry for Life, which is geared towards the detection of CD4 cell counts. So, with no further ado, I will let Professor Robinson tell you part of his story.
Dean Williscroft and Professor Knoll and Mr. Barlin and honoured guests, thank you very much for the wonderful introduction and the opportunity and honour to deliver this talk on the celebration of Wallace Coulter's 100th anniversary of his birth. And what I decided to do was to try to give you some insight into what it was like while Wallace Coulter was coming up with ideas and how he succeeded. And I think there are a few people in the audience who may have been around when Wallace was making those discoveries, but some were not. And so I would like to try to take you back in time. And before I proceed, I'd like to acknowledge the Wallace Coulter Foundation for their work over many years in supporting biomedical research. I'd like to acknowledge Bob Auer, who used to work for Coulter Corporation, who assisted me with a lot of the material that I obtained, and many colleagues who have helped to provide the information that I'm going to show you today. Now, I titled my talk to discuss several aspects of Wallace Coulter's life, and you heard already from Mr. Barlin that he moved around the world through Asia as a salesman for General Electric, and he learnt a lot about the culture of Asia, and that really changed his life to a large extent. And during his time as a salesman and a serviceman for General Electric, servicing X-ray technologies, he was visiting a great number of hospitals and clinics, and there is no doubt in my mind that Wallace Coulter started to understand the complexities of delivering healthcare. And he constantly was thinking about how it could be done better. And the last couple of general things that I would say is that apart from learning about the culture of other countries, he also became an avid book reader and a connoisseur of tropical fruits, thus the farm that Wayne mentioned, and of course a very adroit businessman. You've already heard about his background in education and early jobs, so I'm not going to go into that, but I will say that while I'm going to be focusing mostly on Wallace Coulter, it's very clear that Wallace and Joe Coulter were extremely close, and a lot of the decisions that were made together. And you heard that he did attend Georgia Tech. I seem to think that perhaps he was too busy to finish Georgia Tech. He was not a person interested in awards and certificates in any way, and I think he was focused on the bigger things of life. I think he had a vision that he wanted to achieve, and spending those last few years in college, not unlike a certain Bill Gates and others who forgot to finish their education, and look what happened to them. So Wallace Coulter was a different thinking person. He was extremely intelligent, and Wallace knew that what he needed to succeed, he could learn out there in the world doing the things that he wanted to do. But he was an extremely unassuming person, and he almost always sat in the back seat so that he could observe how other people were responding. Now, let's start in the 1930s. Many of you won't remember, I'm sure, what happened in the 1930s, but this was the time that Wallace Coulter's mind was being formed. Polaroid photography, the zoom lens, the light meter, the parking meter, the radio telescope, the electron microscope, FM radio, something very close to Wallace's heart, was developed. Nylon, radar, the photocopier, the jet engine was built. The ballpoint pen was invented. The cell counter, one of the early cell counters by Moldavan, which I'll mention again, was published. LSD was synthesized. And Teflon, freeze-dried coffee, and helicopters was invented. This was the state of mind of the world that Wallace Coulter grew up in the 1930s. And as Mr. Barlin said, that he worked in a radio station as an engineer and also an announcer, he developed his skill in electronics during this time, and he was interested in building amplifiers. He thought that he could build a better amplifier than anybody else. And in fact, he spent many, many years building amplifiers for hi-fi, which is what it used to be called. So he starts to understand the implication of technology and industry. And when he joined General Electric, it's very clear to me that he joined because he saw a worldwide technology company. And he did not join General Electric to sell X-ray instruments. He joined General Electric to understand how a big company operates. In 1939, he boarded a ship and went to Asia. And I'll come back to that story. So we just talked about the inventions in the 30s. In 1934, Andrew Moldavan published a half-page paper in Science. Wouldn't that be nice if we could do that today? And it described a cell counter using electrical signals and light. I'm sure that Wallace Coulter looked at this at some stage. The next year, there was a very interesting paper that talked about the impedance of blood. Perhaps Wallace read this physics-driven paper and started to think about the possibilities of measuring impedance in blood cells. And in case you were wondering, General Electric released the car radio in 1938, and this is the police version of a car radio. And this is the year that Wallace joined General Electric. Now, it's a portable car radio, and in the trunk was the rest of the components. And from my reading, that several police cars had lots of problems with this because it had huge batteries and generated vast amounts of heat because it had dozens of valves, which didn't seem to work too well on dirt roads. But this was the latest technology that was there when Wallace Coulter joined GE. In the 40s, it was very tumultuous. You know what happened. War was declared. The Battle of Britain in 1940, the Japanese attacked Pearl Harbor and brought the United States into the war in 1941. The Manhattan Project started in 1942. Ballpoint pens go on sale. FDR dies in 45. ENIAC, you've all heard of that computer, was built in 1945. The microwave oven was invented. We drop an atomic bomb on Hiroshima and Nagasaki in 45. The sound barrier was broken in 47. Polaroid cameras were then invented, as they had already had the film from the previous decade. The State of Israel, 1948. George Orwell published 1984, something I'm sure every person in the room read when you were in high school. The transistor was invented. Papanicolaou developed his diagnostic codes for cervical cancer in a famous publication in 1941. Gurkha developed a machine to count mine dust, and he was a mining engineer who was taken over by the military during the war because they wanted to use it for looking at biological particles. Fluorescein was first conjugated to antibodies in 1941 by Coons and Kaplan. That is the environment in which Wallace Coulter started to think about developing technologies. So, what was happening in Wallace Coulter's life during this time? He spent 2 years travelling in Asia. He escaped from Singapore. He took the long route home, as Wayne said, travelling from GE office to GE office. He spent 3 months travelling around India, learning about the culture of India. He spent some time in Cape Town, South Africa. He resigned from General Electric actually while he was on the road. How many people would do that? Only a highly driven individual who knew exactly what he wanted to do when he got home. He started to develop business interests. In 1946, he moved to Chicago, worked for several companies, including Raytheon, and he started a company called Coulter Amplifier Company, and he started to build hi-fi amps because, as you know, valve amplifiers are beautifully beautiful in their sound reproduction. And even today, many people buy valve-based amplifiers in preference. He designed a pasteurization system for milk, and he patented that. And in 1949, he submitted his patent for an impedance-based cell counter. I mentioned that Coons and Kaplan labeled antibodies with fluorescein. This was the very first time that anyone had used a targeted antibody. So this is a very important technology, and this was happening right at the time Wallace was being imprinted with these ideas. Here is Gucker's particle measuring machine, and Gucker was one of the first people to use a photomultiplier tube. And the brightest light source at the time was a Ford headlamp. At this time, Wallace Coulter drew these drawings on your left, and those were the handwritten drawings of his patent submission, which was called electrical sensing zone method. We moved to the 1950s, and what was happening? Colour television was introduced. Winston Churchill became Prime Minister again in the United Kingdom. Queen Elizabeth was crowned after King George VI passed away. The seat belt was introduced, but probably only in luxury cars. You had to be rich not to die in a car crash in those days. Polio vaccine was introduced in 1952. Mount Everest was summited in 1953. The 4-minute mile. Disneyland opened. McDonald's Corporation was started. Rosa Parks refuses to give up her seat on a bus. Sputnik is launched by the Soviets. NASA was founded, and Castro becomes dictator of Cuba. This was the environment that Wallace Coulter began his business. Coons and Kaplan developed fluorescein and expanded that, and developed fluorescein isothiocyanate, which wasn't quite as dangerous as using phosgene gas to create the fluorescent molecule. Melors developed a scanner for cervical cancer. Mary Lou Ingram, who you saw on that video, started to understand the abnormalities of blood cells during radiation. And it was Mary Lou Ingram in the 50s who first contacted Wallace Coulter and called him on the telephone and said, "Mr. Coulter, will your counter tell me the difference between abnormal and normal lymphocytes?" And he said, "I think so." And so she arranged to purchase an instrument, and Wallace and Joe Coulter put that Coulter counter into a trailer, built a device to power it, and Mary Lou Ingram, drove a car with that trailer out to Nevada and Colorado, and she measured the blood samples of hundreds of mine workers who were working in uranium mines, and published seminal papers on this publication. So Mary Lou Ingram drove that vehicle, and Joe and Wallace personally outfitted that trailer with a Coulter counter. And that shows the dedication that they had. In 1953, while Wallace Coulter was trying to market his Coulter counter, Watson and Crick defined the structure of DNA. Crossland-Taylor developed the sheath flow principle, which is basic to all flow cytometers and a lot of cell counters. Marvin Minsky invented the confocal microscope at MIT. And it's interesting if you look at the state of knowledge of leukocytes at that time, and I'll show you, very little was understood about leukocytes in 1953. 1954 was the first report that cigarette smoking might cause cancer. And in 1956, the first, another patent driven by Parker to develop another cell counter. So Wallace's life, what was he doing? He settled in Chicago. He was building things in his basement laboratory. He was also working full-time. He submitted his patent for sterilization for milk, and he was trying to work out how to build his commercial systems. His patent was finally issued in 1956, but it was a year or so later that Joe and Wallace put together another patent, because one of the difficulties with the Coulter counter was the requirement for precise volume measurement. And they couldn't really do it in those days. Didn't have micropipettas that we have. And so Joe and Wallace developed a dilution device which they subsequently patented. And it's interesting because the combination of those two patents really made the Coulter counter unique and reproducible. The Coulter company, he still thought that he was going to sell amplifiers. He was also building amplifiers for other technologies, but he was very interested in industrial applications. And it was also in the 50s that he started Coulter Electronics. So he was fascinated by amplifiers, and he really wanted to build an amplifier company. I think he wasn't certain whether or not his technology would be successful, and so he wanted to have a backup. He published a single paper in his entire life. How could possibly be successful if he only ever published one paper? And he was smart enough to get very good people to test his equipment, and I'll explain that in a moment. Here's the paper that Coons and Kaplan published, where they worked out how to manufacture fluorescein in a way that didn't kill everybody who manufactured it. And this was subsequently driving the clinical business of imaging, where people started to now use antibodies to target cancer and to look at DNA in particularly. And it was in 1951, Wallace Coulter wrote to the Navy and said in his letter, "I have come up with a great idea for measuring cells accurately. And with all this work that's going to be needed to track radiation, particularly if we get bombed by the Soviets," because this was the thinking at the time, he said, we should be prepared and we should really build lots of these instruments. So the ONR funded him to build 2 instruments. At the same time, Robert Mallours was building instruments to look at the changes of DNA in cancer cells, looking at the fact that DNA changed, and they were starting to understand that you could measure these changes and therefore identify abnormal cells. This is the paper that Mary Lou Ingram published when she started working on the blood of cyclotron workers who were overexposed to radiation, and Crossland-Taylor's publication on the sheath flow principle. All of these were, as I said, fundamental to the success of all cell analysis technologies. And this is an interesting paper I found in 1953 in the Bulletin. And I read this paper and I was rather shocked at the lack of understanding and knowledge of white blood cells in 1953. In 1973, 20 years later, I did the first immunology course as an undergraduate at my university. And in the honours class that I did the next year, the students were divided into groups, and we had to debate the fact that small lymphocytes were more important than large lymphocytes. And that was the sort of level of knowledge at the time. Now, I wanted to address the issue of Wallace Coulter and paint. And you heard about a little discussion in that video about paint. And there's all sorts of stories about paint. And some of the stories say that he used paint to develop his ideas, and that blood was a good substitute for paint on a cold evening when the paint solidified. Was he looking for a better way to analyse paint for the Navy? Nobody really knows. But I decided to do some study to see if it would be possible to look at paint or paint components on a Coulter counter. What would Wallace have been thinking in 1949, 1950, 51, 52, in that time when the Navy was building vast numbers of ships, when there were all sorts of interests in industrial technology? So I looked it up, and of course paint is coloured by pigments, and those pigments are powders, and the powders have different sizes and shapes. And the idea was that you couldn't accurately measure these powders. In fact, it's a huge, and was a huge, industrial problem in industry. And Wallace was very aware of that, and you heard on that video several people involved in that. So I can just imagine Wallace thinking, how can I work out how to count those particles better than somebody else, more accurately? And I looked up the literature to find out what was the size of particles, and you can see they go from 100 microns, 500 nanometers, 500 nanometers, 1 micron, well within the range of a Coulter counter. Coulter counter's range, advertised in 1956, was a half micron up to 20 microns or so. He even had systems he designed to go up to 800 microns. So Wallace could easily have measured these components. Now, what is the story about the Navy? Well, here's a Navy ship, and it's grey. And the question is that the Navy was worried about, from my understanding, was how do you get enough paint that looks the same when you paint something very big? Now, that little box represents all the walls in your house. So if you were to pick one colour and buy the paint for it, that's about how much paint you would want. And the Navy didn't want their ships to look like this. They wanted to be able to buy huge batches that had the same colour, and that was a well-known problem at the time. And I can just imagine Wallace Coulter working out, well, the reason those greys aren't right is because they didn't have the right number of particles. They didn't calculate them properly. And I went and looked again at the literature, and there's a lot of discussion about measuring paint in the 1950s using Coulter counters. So this is a paper that discusses that. These are the environments that Wallace lived in at the time, and he got sick of working in his basement, moved actually twice, and he moved into this building. And one thing you'll notice about this building is that there are railway tracks behind it. And as they built and calibrated Coulter counters in 1956 and 1957, 58, 59, they had to stop their calibrations every time a train went by because the building rocked. And so that was Wallace's life. And I've shown already these handwritten drawings, but this is a bigger picture of them. These were no doubt drawn by Wallace Coulter himself. He had a knack for advertising, and he did things in a very simple way. And this is his only publication, which was in the National Electronics Conference in October 1956. And it's a very interesting, technically detailed publication. But what he was smart enough to do was go to the very best person to test his prototype. So he went to the National Cancer Institute. The National Cancer Institute in 1956 was only a few years old, 3 or 4 years old. And he went to George Brescia, and the Navy provided the two instruments that they had paid for, and George Brescia published this paper, which fundamentally said, look, this instrument is so much more accurate than hand counts. This is the way to go. He started advertising in many journals in the 50s and 60s. This is a paper that I'm also sure he wrote, because the technical information in this paper could only come from Wallace Coulter, and there were only 2 or 3 people working in his company at the time. Here's another advertisement in 1957 where they talk about actually measuring dairy products, oil globules, and size of particles in paint. And this is a typical advertisement at the time. And he had competition. This is from Crossland Taylor, who you remember developed the sheath flow principle, and in the United Kingdom they started a company to compete with Wallace, selling an instrument that used light to measure particles. And of course Wallace had tried that and it didn't work very well. He started Coulter Industrial Sales because he recognized that there were many facets to particle counting, and he wanted to get out there into industry. So we move on to the 1960s. What happened in the 60s? The laser was invented, the birth control pill. The Berlin Wall went up in the 60s. JFK gave his famous Man of the Moon speech and the first James Bond movie. And Johnny Carson took over the Tonight Show. JFK was assassinated. The bullet train started in Japan. Nelson Mandela was sentenced to prison in 1964. Winston Churchill dies in 65. We went to Vietnam in 65. And the first heart transplant in Cape Town. Christian Barnard. 6-Day War in the Middle East. Robert Kennedy and Martin Luther King both assassinated. And then we ended that decade with the moonwalk, and one of the more important things for our children, Sesame Street, was first aired. And in the world of science, a critical paper that actually started the process for the invention of the flow cytometer was published by a pathologist in 1962, and I'll tell you that story in a moment. Lou Kamensky, who worked for IBM and whose neighbor was a pathologist. The pathologists seemed to be contaminating the world of technology development, challenged Kamensky about trying to measure cancer cells, and so he started looking at developing systems in IBM. Acridine orange was invented or discovered in 64. The inkjet printer, another critical component in the invention of the cell sorter, was invented in 65. Mac Fulwiler developed the cell sorter in 65. And then Kamensky built an instrument which ended up being Technicon's blood technology, sold by IBM to Technicon. The cell sorter for fluorescence-activated cell sorter was developed by Herzenberg, and Mac Fulwiler finished his invention without his PhD, went off and then got his PhD and went back to Los Alamos. And in Europe, Wolfgang Goethe developed the impulse cytophotometer, which was one of the first flow cytometers that measured fluorescence. So what was happening in Wallace Coulter's life? In 1961, they decided that they would move the company to Florida. Rumor has it he looked everywhere, anywhere that was hot, anywhere that was different from Chicago. And in 1961, Coulter Electronics claimed they had 1,500 Coulter counters in the field. Larry Kuntz, who you saw on that video, was hired in 61, and in 61, Coulter Corporation, Coulter Electronics at the time, had sales of $3 million, which I think is phenomenal at that time. He started a large industrial base, and he was heavily advertising in journals. He started clinical chemistry and hired Harold Crews in 1966, and he really pushed his industrial business. And in 1968, he developed the Coulter S. This is an advertisement from a 1961 journal showing fine particle analysis and breakthroughs. This is Lou Kamensky in IBM Watson Labs developing pattern recognition. It was Lou Kamensky who was the first person to develop the algorithms for character recognition. And he did this, by the way, to build an automated telephone system. And guess what the internet was invented for? And in CERN, to do a telephone directory. In 1962, this was the state of art of a Coulter counter, and this is the inside with multiple valves. You didn't need to heat your labs in those days. You just turned on the Coulter counter. It's another advertisement from 1962, and this is the paper that started the revolution. What happens when a pathologist gets hold of an instrument that has knobs on it and doesn't understand what those knobs mean? Well, this pathologist by the name of Lushbo used a Coulter counter and measured blood and decided there were two distinct populations of blood based on impedance. An engineer in the lab looked at that and said, it doesn't make sense to me as an engineer. I know nothing about blood, but it doesn't make any sense that you could have two populations of blood based on impedance because of this, this, and this, some physics. That paper in 62 started a bunch of people to talk, and in 1964, Richard Sweet developed the inkjet printer, and he gave a talk that Mac Fulwiler attended in 1964. And Mac Fulwiler saw that what Richard Sweet had done was to build a high-speed printer because he couldn't put a pen on paper and run the paper fast enough to get up to 200 kilohertz in frequency. And so he said, well, why not fire the ink at the paper? And we'll use a piezo crystal based on frequency changes of voltage of our signal, and that will give us the chart. So we built a chart recorder. And Mac Fulwiler saw that, and he asked the question, could we put cells in those droplets? And this guy published this paper that basically said there's two populations of blood. If we built an impedance-based instrument that was an inkjet printer that could separate them, maybe we could build a cell sorter. At the same time, Wallace Coulter built the most enormous Coulter counter ever built. This is a Coulter C. This consumed 20,000 watts of power. It had hundreds of valves in it, and this was a behemoth of an instrument, and it was built for industry. So not many of those were built. I mentioned already about acridine orange. You just heard about Richard Sweet, and Mac Fulwiler now trying to build a cell sorter. And with Marvin Van Diller, they did build that cell sorter, they patented it, and they were the first people to develop droplet counting and droplet separation, and they used impedance. And Kamensky went on to separate cancer cells using a microfluidic device, which he built in 1965. Microfluidics is not that modern. This is the instrument that turned out to be the Technicon blood analyzers. This was built by IBM. IBM decided they didn't want to be in the blood business, and they moved out of that business. A lot of technology using measuring particles at the time. Lots of publications, and even publications in blood saying that you could measure different types of particles, and something that is a more recent interest, I think. And then computers came along, and this is the first publication that I could find of matching a computer to a Coulter counter is 1968. In 1968, something very interesting was happening. The Coulter S blood counter was developed. When they started building it, it was all valves, and it looked bigger than the Coulter C that I showed you a little while ago. But, you know, the transistor had been invented, and the integrated circuit had just been invented. The total supply of integrated circuits manufactured in 1962, 100% of them went to the military. But by 1968, production had increased sufficiently so that only 37% went to the government space and space program. Do you know that 5% of all integrated circuits built in 1968 were purchased by Coulter Electronics? And that made the success of Wallace's biggest instrument, most successful technology, the Model S hematology counter. And it had over 1,200 semiconductors in it. So in 1969, about the same time, Dietrich and Goethe published the fact that they could measure fluorescence in moving cells, and Herzenberg started to build an instrument that he was interested in measuring lymphocytes, and he invented the term fluorescence activated cell sorter. So what was happening in cell sciences in the 70s? This is a fascinating story, because this is the start of Becton Dickinson and Beckman Coulter as competitors in the field of flow cytometry. Becton Dickinson was negotiating with Los Alamos because that's where Mac Fulwiler was, and that was where the technologies had been developed. But Wallace Coulter, being a savvy businessman that he was, snuck in the back door and signed the Los Alamos team 3 weeks before Becton Dickinson. And that just shows you how smart Wallace Coulter was, that he was able to do that. Lots of things developed during that time. 1975, Kohler and Milstein published monoclonal antibodies. By the way, that publication is a mere letter in Nature. It's not really a real paper. So one looks at that and sees how amazing it was. And of course, Cambridge forgot to patent it. So that's good for the rest of us, I suppose. Okay, so what was happening in Wallace's life? He went to Los Alamos knowing that the Coulter counter had been used and abused by Los Alamos. So interestingly, he saw that they were not purchasing instruments. They were building their own. So he sued them. And Wallace protected his technology. But in an agreement, it turns out that after he sued them, he actually made friends with them and then locked them all in as consultants and hired them and started a company called Particle Technologies, made Mac Fulwiler the president of that, but Wallace owned that company lock, stock, and barrel. In 1974, they started selling big instruments, and in fact, they wrote to NIH, and NIH said, we want to fund it, but we don't know what to call it. And there's a very interesting letter that I have that talks about what should we name these instruments. Mac Fulwiler actually started Particle Technologies but in 71, but in 75, he decided to leave. And he didn't just go anywhere, he joined the competition. And that really annoyed Wallace Coulter, and I'll show you what that led to in a moment. But Ortho Diagnostics was moving quickly. They had purchased Kamensky's company, and they started making monoclonal antibodies. They were the first commercial company to make monoclonal antibodies. For those of you who remember, the very earliest monoclonals that you could buy were called OK antibodies, OKT4, OKT8, and they came from Ortho. And Kamensky built a company called Biophysics at this time. It is thought to be the first commercial instrument of any nature containing a laser. And there were lots of discoveries at that time. The concept of separation of lymphocytes, monocytes, granulocytes by light scatter was only just discovered in the 70s. And there's lots of papers in that area. And in 1971, this very interesting discussion between Becton Dickinson and Coulter. And here's a memo from Dick Schwartz, who was head of Becton Dickinson at the time, talking about whether or not they should invest in Stanford. Was it a good deal to invest in Herzenberg or not? And were these instruments of any value or not? And if they decided to build flow cytometers, how would they do it? And this is fascinating. They didn't know where to buy a laser from, so they decided to buy Lou Kamensky's ortho instrument and take the laser out of it and use that for their own technology. That seems to me to be a sort of fascinating look back into the minds of engineers at the time of the early 70s. They calculated the cost of building a flow cytometer was $15,000. Wouldn't that be great if that's how much we would pay for them today? So then they decided they didn't think they needed Stanford, that they'd do very well without them. And suddenly Wallace Coulter popped out of nowhere, purchased everybody at Los Alamos, and 3 weeks later, Becton Dickinson signed with Len Herzenberg, and of course, the rest is history, because Len Herzenberg then developed the fax instrument. And interestingly, it was Len Herzenberg who went on sabbatical to Cambridge University, and he went to the laboratory of the discoverers of monoclonal antibodies, Kohler and Milstein. Milstein's actual lab at Cambridge. And it was Len Herzenberg who made the term hybridoma, because they didn't know what to call that funny combination of a lymphocyte and myeloma cell. And Los Alamos was now using a flow cytometer to sort chromosomes, and that was the beginning of the Human Genome Project, because it was flow cytometry that separated all the chromosomes so that they could analyze them. And then in that time, Wallace's competition was pounding out instruments, and in 1974, Becton Dickinson announced that they would demonstrate the first commercial cell sorter. Wallace Coulter got hold of this note, and he was livid. He walked into Bob Auer's office. He said, "I don't care how you do it, but you will have a cell sorter on the bench at that FASB meeting in Atlantic City in April." And this was about 2 months earlier. And sure enough, Beckman Coulter demonstrated the TPS-1, the two parameter sorter version 1. And that is a picture taken at that show in 1974. And this is a letter from the NIH trying to work out what to call this new device called a flow cytometer. And it was Coulter that was trying to build these, and in fact they built one called the EPIX-2, and this was delivered to Dr. Chet Herman in the NIH in 1974. And it was the Hemolog D that was developed out of Kamensky's instruments. And I mentioned that Mac Fulwiler in 75 decided to leave particle technologies. And the day that he announced he was leaving, Wallace Coulter placed this piece of paper on your left on Bob Auer's desk. And in typical Wallace Coulter fashion, it just had one word on it. And it said, faster. He did not want Mac Fulwiler taking his ideas to Becton Dickinson and beating him. He was an extremely competitive individual. And on the right here, on your right, you'll see the paper that Milstein acknowledges that it was Len Herzenberg who, in 1975, termed the term hybridoma. And in 1977, Wallace Coulter was sorting chromosomes using the TPS-1, and They were looking for B cells and T cells. Of course, it was T cell rosettes in those days, which if you're an older immunologist, you will know what a T cell rosette is. I was speaking to a younger audience recently and none of them had a clue what a T cell rosette was. Anyway, that shows you that you can differentiate cells that attach sheep blood cells. And then in the 70s, Coulter really drove their business in flow cytometry. And so we moved to the 80s. John Lennon was assassinated. Ted Turner established CNN. And AIDS was identified in 81. IBM releases their personal computer. Sally Ride becomes the first American woman in space. New Coke hits the market, and I remember that because I was moving from Australia to Michigan, and I had a lot of friends who asked me to carry cases of old Coke to America because people didn't like New Coke, and of course it failed. The Space Shuttle Challenger exploded, and I was sitting right here in MedSci 1 when that happened. I have a very clear memory of that happening. And Chernobyl, a nuclear disaster. And DNA was first used to convict criminals in 1987. And of course, the Berlin Wall, which we mentioned a little while ago, was formed, crashed in the 1980s. And in Wallace's life, his life was dedicated to building lots of different instruments. He built his multi-sizers, his entire EPIX line, and the Q-Prep, which was revolutionary in 1986 for rapid preparation of staining of blood cells. And in the 1990s, we know a little bit about Bexar and the various new instruments that were developed by Coulter Corporation. And by this time, of course, Wallace was quite elderly and not particularly well. But in 1995, they made a big decision, and I'm sure Wallace had a lot to say about this when they acquired Immunotech in France as a major supplier of their monoclonal antibodies. And in 1997, as Wallace was really fading, Coulter was acquired by Beckman, and of course Wallace Coulter passed away in 1998. Now, I have not given you all the stories of things that happened during this time. I have biased what I've said towards what was developed more by Wallace Coulter and his colleagues, and there are many other companies and individuals that I haven't mentioned that have done amazing work, and my apologies for not talking about those. But my goal was to really give you some idea of how and why Wallace Coulter, over the many years that he spent driving his company, building a private company up to a $2 billion company, building technologies that really impacted people's lives. And I hope that I've given you some insight into how Wallace must have felt as he made his discoveries that have impacted all of us. Thank you all very much for your attention.
So just our final closing remarks will be from Vice President Steve Forrest.
Well, I'm Steve Forrest. I'm the VP for Research here at the University of Michigan, and I want to thank the Coulter Foundation for their terrific partnership over the years. It's been a terrific partnership from many standpoints. Obviously, there's the fact that they are in partnership every day with our Office of Tech Transfer. They have spurred on a few companies. Actually, we have, I don't want to hold people up too much today, but we have over in Celebrate Invention, we have one of our Coulter companies, it's Shu Takayama's company, Phasic. Also, one of the guys working right out of my office, Brian Folks, has one of the companies, which is Histosonics. Coulter also has been extremely helpful with the state of Michigan. In partnership with them, we have 2 MTRAC programs, one between in the biomedical sciences and the other in transportation. I'll probably have a little bit more to talk about transportation when we go next door. But overall it's had quite an influence, but it doesn't work in a vacuum. There's a substantial ecosystem here that we've been building up over the last decade or so to spur on these types of partnerships and to really encourage our faculty to get out and start companies. And this is just one more piece of it, but it's a very essential piece, and it brings engineering and medical sciences together in ways that are quite unusual and quite productive. Just to give you a couple statistics, again, I'll talk a little bit more about this next door, but right now we're a $1.35 or so billion research enterprise here. That's nice. We like big numbers. Actually, that's very nice because we like really big numbers. But a piece of that is 8% of our external funding comes from industry. And the NSF national average for research universities is 5%. So we have, and by the way, when I started here about 8 years ago, we were pushing 5% from the low end, from about 4.5%. So something really big has changed, and I think what happened is that we have built this ecosystem at the University of Michigan that encourages faculty not only to start their own companies, but to work with other companies and so on. So again, you know, when I also, pretty close to when I came here, we got our first Coulter grant. And I became aware of it quite early and thought that this is exactly the sort of research that we want to do here. And I've had actually a fairly long relationship with Doug Knoll here, who's been one of the champions of this whole business, and we've gone off to China and had many a good drink over there too. But we've talked about how to improve our methods here, and so I want to thank Coulter. It's quite an auspicious occasion today because it's Wallace Coulter's 100th anniversary. I am told we have some cake back there in celebration. So I think with that, I will just let everybody enjoy the rest of the afternoon. I hope a lot of you go next door, but we're really looking forward to continuing with the Coulter Foundation in the future. We're very proud that we're a recipient of that, and we want to have a leadership role in making these things work in the way that Coulter and ourselves both believe they should. So thank you all very much, and I invite you to celebrate in the back.
I worked for a venture fund, for an investment bank. I had my own consulting company, and I witnessed many failures. At certain point, companies failed, project failed.